Good morning. I'm Susan Ellenberg from the University of Pennsylvania, and I'm very happy to be at this conference. It's been very, very interesting. Before I start, I would just like to say the issue of oversight. I think most aspects of oversight of research that we have now has been put in place because something awful happened at some time or other, and people had the view, whether it was HHS or Congress, that this is never going to be allowed to happen again. No matter what we do, these kinds of things are going to arise from time to time, and it is difficult to pull back on oversight mechanisms for that reason. So I think that's just something that we have to keep in mind when we talk about oversight. So I'm supposed to talk actually about focusing on pragmatic trials, and so I thought it would be good to remind people that this is really not a new concept at all. This concept was actually first introduced by two French statisticians, Schwartz and Leluc, in the 1960s, who made the distinction between explanatory and pragmatic trials. So explanatory trials are trials where the purpose is to answer very precisely a scientific question, controlling heterogeneity, making everything as homogeneous as possible so that you can isolate a treatment effect. And these are the sorts of trials that are done in a regulatory setting to evaluate new drugs and other kinds of new medical treatments. A pragmatic trial is to answer a practical question. Which approach should we use? The idea is to conduct a trial under real-world conditions with the intention that the results be highly generalizable. And I'm going to be focusing on clinical trials in this talk, not observational studies. This kind of took hold particularly in the UK, and in the 1980s there was a huge push over on the other side of the pond to do what they called large simple trials to answer important healthcare questions. The idea was that there might be what we would consider small benefits, but because of the huge populations affected by these diseases, we could save a lot of lives if we found even a 1 or 2 percentage point improvement in survival. And so we talked for a while about large simple trials, which are basically large pragmatic trials, eligibility based on the uncertainty principle, as Sir Richard Peto calls it, meaning that the eligibility criterion is simply that the physician and the patient together are not sure whether or not this was a good treatment to use for the patient and the condition that he or she is in. Minimal restrictions on care other than the assigned treatment, basically do what you always do. Rigorous attention to control bias by focusing on the control of systematic errors rather than random errors. And that relates to another aspect of oversight, monitoring, which I will mention in a bit. So the idea was to get a reliable assessment, a very reliable assessment of the effective treatment in a real-world setting. And four big trials, increasingly large, were conducted by the British group. These were the international studies of infarct survival. The first one was reported in 1986. The fourth one was reported in 1995. They were complicated designs, but they identified small but real improvements in survival in people recovering from myocardial infarction. And as you can see, these trials were quite large. So the size of the reductions that they found would never have been seen in a smaller study or in any kind of an observational study that one could have relied on. So the push for these kinds of trials was to make them as simple as possible. It's kind of hard to imagine for those of us who have been doing clinical trials for a long time, but the data collection was very limited. The baseline data was collected by phone at randomization. The physicians putting their patients onto these trials didn't have to fill out any baseline forms. There was a single-page form. In fact, I think they like to say that it was a half-a-page form that was collected at hospital discharge, and no eligibility criteria other than if they didn't have any contraindications or allergies or something like that to the given treatment, just entry on the basis of the uncertainty principle. So those were done, and things kind of, people kind of forgot about them. I remember when I was working at NIID in the late 1980s and early 1990s, we had some discussions about whether, if there were a candidate AIDS vaccine, could we do a large simple trial of an AIDS vaccine, and Tony Fauci asked me to actually do a presentation on that once at NIH, but he wasn't very optimistic. He used to call them large expensive trials, not large simple trials. But now there is an increasing interest in pragmatic trials, primarily in two areas: comparative effectiveness studies, which Ruth has talked about, and quality improvement studies, where the idea is we would use randomized trials instead of making arbitrary judgments to make decisions about optimal care management. So with all of that introduction, what about oversight? Well, I'm going to talk about briefly three different types of oversight. One is the IRB, or Institutional Human Research Protection Programs. I'm not going to say more about that, very much about that, because there are many more people in this room much more knowledgeable about how IRBs work than I am. I'm going to talk about ongoing monitoring of data quality and the use of data monitoring committees. And so there certainly has been increasing discussion, as we have just heard, about what types of oversight mechanisms are needed for certain types of pragmatic trials. So let's starting with the IRB. Here's where we have huge amounts of debate. And so when argument goes, if a hospital doesn't need to get IRB review, if they want to decide to switch brands of antibacterial soap, why should they need IRB review if they want to do a study comparing two brands of antibacterial soap or two types of dispensers for antibacterial soap? Whether they would ever do such a study, want to do such a study, I don't know. But under the current circumstances, some IRBs might say you need to get IRB review for this. But what if instead of soap, the issue is whether to adopt a new diagnostic assay as a routine procedure? This is the kind of arbitrary decisions that hospitals would make all the time, but people might wonder whether there's really evidence that the expense of this new procedure is going to add to people's, is going to be a benefit ultimately to the people treated at the hospital. And then what instead of this new assay, the issue is the duration of a dialysis interval, as is the question in a study that I'm currently involved in, as is Dr. Jaffe, or the percentage of oxygen that should be given to newborns with respiratory distress, or the tidal volume in people with ARDS. So where do we? I think this is where Dr. Metnikoff was going, as to where do we draw this line? Does the endpoint of the study make a difference if both regimens are considered standard of care? And then does it make a difference if it's an individually randomized or a cluster randomized trial? And I'm going to say more about cluster randomized trials at the end. I should say that there will always be debates on these issues. There are still people, thoughtful, knowledgeable people, who argue that randomized trials where you don't keep each patient apprised of what the current comparison is of the treatments is unethical, that patients should know exactly what's going on. That is a very much of a minority view, and I think most of us know we wouldn't be able to do randomized trials if we did that. But so there are always differences of opinion, and the fact that we aren't going to get 100% of agreement is not a reason not to try and come to some judgment where people can make their own decisions and accept that not everybody will agree. So I want to say a little bit about monitoring of study conduct. The quality of study conduct is always important. If the study is not conducted well, you can't believe the results, or it might produce results that are wrong, even if people believe them, and so there are ethical considerations there. Many people are I shouldn't say pushing, that almost sounds pejorative, are arguing that most important monitoring can be done centrally, not requiring on-site checking of data accuracy. That's become a huge, huge business, and there are many of us who believe that the costs of clinical trials, this is the one area where we could probably have the biggest bang for the buck in reducing costs of clinical trials without really damaging the validity and accuracy of trials. Errors in data entry, there are a lot of studies that show, especially now where we have electronic data entry and some checking as people enter data, should be minimal. It should be random. It's not likely to create systematic bias. Most of these errors can be identified by a central review, and indications of quality problems from central review can lead to site visits as necessary. This is traditionally how Many NIH-funded trials have been monitored. There's been much more on-site monitoring these days, much to the distress of investigators and probably NIH staff who have to figure out where the money goes. And I think this is an area that needs to be rethought. It's a big organization, a public-private partnership between Duke University and the FDA called the Clinical Trial Transformation Initiative that's been doing work on this issue. Monitoring the accruing data on safety and efficacy has traditionally been the province of Data Monitoring Committee, as FDA will call it, or Data Safety Monitoring Boards, as NIH prefers to call them. They will monitor the accumulating data from some randomized clinical trials, generally trials with serious outcomes or trials with anticipated potential safety issues. And I think this is not really different for pragmatic trials versus other randomized controlled trials. Not all trials require a data monitoring committee. I personally think we've gone overboard in the assignment of the requirement of data monitoring committees for a large number of trials. But probably most trials that are going to call themselves pragmatic trials will require these because they are going to be addressing public health questions and will probably be fairly large. So probably most people in this room know what a data monitoring committee is. It's a group of experts who, without other involvement in the trial and with no relevant conflicts of interest regarding the trial outcomes, who will review the accumulating regularly and make recommendations to the sponsor and investigators regarding any possible safety issues, emerging quality issues that may endanger the validity of trial results, and obviously to see whether the trial question's already been answered by the results that have come in so far, and whether the trial should terminate early. I think the principles and practices for these committees for pragmatic trials are going to be mostly the same as for any trial that requires a data monitoring committee. There will need to be a pre-specification of the statistical criteria for early termination. There'll need to be regular review of the data on the safety and quality of study conduct. You'll have a multidisciplinary group that will avoid major conflicts of interest, and the confidentiality of interim results will be maintained. Those are pretty standard. I think there are a couple of special issues in pragmatic trials that I do want to mention, one having to do with comparative effectiveness studies generally, and one having to do with early stopping guidelines. Comparative effectiveness trials will be comparing one standard treatment approach to another, and in any trial like this, you have to be very cautious in interpreting a finding of no difference. No difference could really mean no difference. It could mean the outcomes were too variable, or the study size was too small to permit the detection of an effect that might have been important. It could mean the trial quality was poor, lots of noise, diluting the observable effect. This is an issue in any active control trial, but it may be enhanced in quote, pragmatic trials, where there will likely be more heterogeneity than there is in many active control trials. And so there's an opportunity for more noise. And that is something that an oversight group like a data monitoring committee would have to be aware of. The criteria for early stopping, I think, requires some deep thought for these kinds of pragmatic trials. I think the criteria probably will need to be more conservative than we usually set up for in trials. Typically, in the US anyway, we will set up an O'Brien-Fleming stopping boundary with an overall p-value of 0.05. In a pragmatic trial where you are comparing two treatments that are in wide use, you will have practitioners who are wedded to these treatments, who believe that the treatment that they always use is better. They will need a fair amount of data to persuade them that they should change what it is that they're doing. So you have to say how many zeros before the first non-zero digit in the p-value will be needed to foster change. In the ISIS trials that I mentioned before, I think it was ISIS 2, those trials completed. They did not stop early, and there were, I don't know, 5 or 6 zeros before the first non-zero digit, and there was lots of debate. Even now, if you find people who are old enough, you will find people who will argue about why wasn't that trial stopped early. And the trial leadership argued that they did not stop the trial early because they felt that if the data were not really, really, really strong, you would not get people to adopt these new treatments and therefore save lives. So I think that is a consideration, that we can't just sort of do our routine thing with an O'Brien-Fleming 0.05 stopping boundary in these trials. I would also note that futility analyses, which have become more common in clinical trials in order to conserve resources when it's clear that there isn't ever going to be any difference in a study, will probably not be relevant for these kinds of trials. Again, because if they're really, if the two treatments are really pretty much the same, you want a maximum amount of information to be able to narrow that confidence interval around the possible difference. So I'm going to finish with just a few comments about cluster randomized trials, because these are being used now more widely for pragmatic questions. Again, the concept of cluster randomized trials was introduced first back in the mid-1970s, and they have been used, but they're being used more widely. Cluster randomized trials randomizes units instead of individual subjects. For example, you might have a randomization of different emergency vehicles to different approaches of patient support during transport to a hospital, or randomization of hospitals or clinics to different approaches to infection control, or even randomization of communities to different approaches to prevention of HIV infection. And NIAID is sponsoring several trials like that in Africa. A key design consideration for these trials is that statistical power depends more on the number of clusters, the number of units, than on the number of individuals per cluster. And so I think there are some special challenges for oversight of these trials. The main reason is relatively few researchers have been involved with them, and so they're not familiar with these kinds of issues. You wouldn't want to have a data monitoring committee including people who had never done a clinical trial or been involved in a clinical trial. So that's already one challenge, is finding people who understand how these trials work. Some other issues. In a cluster trial, there are typically far fewer units randomized than you have people randomized in a conventional randomized clinical trial. So you might randomize 50 clinics, and for a major public health question, if you did an individual trial, you would have far more than 50 individuals. So you might have 50 clinics, and maybe you have several hundred people treated at each clinic. You may have more participants exposed to what may ultimately be shown to be an inferior intervention. And so that's something that has to be taken into consideration. The issue of blinding is more difficult when everybody in one site it receives the same treatment, and that could raise concern about interim changes in study conduct, which might differ from site to site. And that relates to the last point that I want to make, which is the intra-site correlation of outcomes, which is sort of the key statistical feature of these trials. In a cluster trial, we have to account for the extent to which outcomes at a given site will be similar and will be different from those at other sites. The higher this correlation, the more sites we are going to need. Just to take the extreme case, if everybody at a given site is expected to have exactly the same outcome, you wouldn't need more than one patient per site. You wouldn't need more than one patient at that site to see what the result from that site was. So you do worry very much about this. If you are randomizing a lot of different sites and one site has a very different patient population that's likely to have better outcomes and they're all getting one treatment, you can see that that's a problem. And so great effort is expended in designing these trials to make sure that the sites are balanced with respect to outcome. It's often difficult to estimate this intra-site correlation prior to the study being started. And that means that you don't really know whether your sample size considerations are way off. If you think the intra-site correlation is 0.1 and at the end it turns out that it's 0.4, your study is going to be way underpowered. So this is something else that a data monitoring committee will have to look at as the data emerge. Similar to in a time-to-event trial, you have to see whether you're having the number of events that you expected to see whether you have adequate power. Here you have to look and monitor this particular statistic. So I will stop there and have time for discussion.